For today, we have starting closest to me, we have Dr. Leif Nalen, who is Division Chief of Neonatology at Nationwide Children's Hospital and Professor of Pediatrics here at Ohio State. And he earned his medical degree from the University of South Florida. Did you go to school with Peter Emby? I believe he was there too.
No, I don't think so.
Oh, different times. Then in the middle, we have Dr. David Lucas, who's director of the OSU Comprehensive Cancer Center Hematologic Malignancies Tissue Bank, and also has an appointment in the Division of Medicinal Chemistry and Pharmacognosy. Thank you.
Sometimes that gets dropped for obvious reasons.
And he has a PhD from the University of Wisconsin. And of course, we've already met William Kisseberth from the College of Veterinary Medicine. And he also has a PhD from the University of Wisconsin. Were you guys classmates, or did you know each other?
I don't know, actually. Possibly.
Well, all three of these researchers have worked with biorepositories, and they're going to talk about how they got involved with that and what the kinds of things questions they were able to ask and answer using biorepositories and their various adventures in the field. So thank you guys very much.
Go ahead.
So I was going to talk a little bit about how I got interested in biorepositories. So I'm a basic science researcher. I have a wet lab, and I do a lot of cell culture stuff, which thankfully doesn't need IRB approval. Actually doesn't need ICOOK approval either, so it's really easy. But I am also a clinician, so I take care of patients. And a lot of the things that we do in the lab, and even in the cell culture dish, sort of point us in directions that might be important for patients. And being a neonatologist, one of the downsides of being a neonatologist is our patients have very small blood volumes, so there's not a whole lot of biological material to get from a patient during their stay in the hospital. And there's not very many of them, so in a year we might get 25 patients that we're interested in. And so it's a really difficult thing to do a clinical study looking at perhaps biomarkers or genes of interest in the neonatal field. And so one of the things that happened at the Nationwide Children's about 5 years ago, they started a repository called the Perinatal Research Repository, which should be on your list of repositories because it's available for all OSU researchers to access and to look at. And what they do is they collect samples and clinical data from mom, dad, and baby, babies that are born prematurely. So over 5 years, they've actually been able to get a lot of data in the database. So in the lab, we have a bunch of cell signaling pathways that we thought might be interesting in human disease. So one of the things that we did is we approached the PRR, the Perinatal Research Repository. We asked them if we could have access to DNA and perhaps serum in some cases to look at studies. And actually, they have a nice process in place, and it was actually a fairly painless process to actually be able to move that forward. So we've actually looked at a series of SNPs in a particular signaling pathway in patients with sepsis. And again, sepsis is seen in about 10% of our patients in the NICU that are born at less than 32 weeks, and that's about 100 patients a year. So again, it takes a while for enough numbers to get in the repository. But since the repository's been going for 5 years, we're actually able to get 23 patients that had sepsis and then double that number that didn't have sepsis that were equal in gestational age and birth weight. We also looked at SNPs in necrotizing enterocolitis, which is an even rarer disease. It's only found in 7% of patients. So we were, again, able to find enough patients and enough controls to actually be able to look at some data and get some preliminary data. We also were interested in BPD and pulmonary hypertension. BPD is a chronic lung disease that patients get, and I'm very interested in pulmonary hypertension. That's actually been my basic research focus. And we're actually able to approach the repository last year and get 150 patients that had BPD, of which about 25% have pulmonary hypertension. We're able to use that data to look for a family of SNPs in the arginine metabolism pathway that I'm interested in. And we've actually published that paper recently. And actually, that paper led us to look at some plasma that they also had in the biorepository. So we were able to do some plasma on some biochemical biomarkers. And we've actually published that paper recently as well. And that data has also served as preliminary data for a grant application that just went in yesterday. So We've been able to utilize the repository in a very small, in a small population that has a very small amount of biological tissue available, and we've been able to do it very well with the perinatal repository. And it's very easy to use. It has a system for, they have a website. You can go on the website, you can put in they have a form there to fill out. The form is easy to fill out. There can be dialogue with people in the Ohio Perinatal Research Network who run the repository about how to make it work. And usually within a couple of days, you hear a thumbs up or a thumbs down whether you can use the specimen. So it's a really easy process, and particularly for neonatologists, it's really nice because it's a way of getting enough sample size to be able to do something meaningful. So, and I'm happy to take questions about how that's worked as we go forward.
So I am a basic scientist researcher as well, and I came about this job, I think, the way a lot of you in the audience have as well, which is just being involved in research initially, and then learning the value of getting access to patient samples or dog patient samples or different types of actual primary cells. And so my research background is just in sort of translational medicine, I guess, is the later aspect of it, but molecular and cell biology as far as how cancer cells function and what's wrong with them, of course, and trying to stop that with different pharmacological agents, usually experimental at that point. And fairly recently, just at the beginning of this year, I was asked to take over the Leukemia Tissue Bank and for the OSU CCC. This facility is one of the shared resources of the Comprehensive Cancer Center. We started this renaming to Hematologic Malignancies Tissue Bank just to reflect the fact that we're trying to capture tissues from all hematologic malignancies, not limit it to leukemia. But we haven't completed that name change yet, so even though that's what's on your agenda, it's still on the website as the Leukemia Tissue Bank, and that's fine. We'll go by that too. But we are not limiting ourselves to leukemia. We're working to get all types of hematologic malignancy samples. And just as the other aspect, I was listening to the questions, and I think that this is an interesting topic as well, and I have certainly a lot to learn as I have really only been in this job for about 8 months or so. But what I was going to say, what I had planned to say, was something about the difficulties of initiating and operating a tissue bank. And Dr. Ramirez covered it perfectly. And if you missed her talk, or if you didn't take very good notes, this is being videotaped. I encourage you to go back and look. She hit on every problem that I have encountered along the way. And the other aspect of this is that I've been at OSU for about 14 years now, and I was actually hired to do research as well as to receive, process, characterize samples that were being sent to us on a phase 3 clinical trial. And this was done under the leadership of Dr. Griever, who's the chair of internal medicine here. And this was where I really learned things the hard way. And if I had someone like Dr. Ramirez 14 years ago, if I had the wisdom to seek out that kind of help, my life would have been much, much easier. So pay attention to what she said, because it's dead on. It's absolutely important. There was a couple of points I was going to emphasize as well, and she did cover these things, but set out your processes early. It's so much easier to postpone a week and set things up properly than it is to jump in and make the change later. We've done that, and it's still a pain. The SOPs that you have for your staff, make them clear. Your staff turns over. I think you'll know as well as I do that lab techs probably aren't paid as well as they should, and they often are temporary, or maybe they're here for a year or 3. The turnover makes it important that your practices are recorded carefully for the next group, including the things that they have learned and developed along the way, because there will always be those as well. For the usual checks on things like freezers, I really sympathize. I have this nightmare where I open up a liquid nitrogen freezer and it's warm inside. That happened to me once, and it's awful. It sticks with you. So it's not just having a good system and a quality check and all that. Have a spare freezer. If you have a freezer that's half full, start the process of getting another one. Don't wait till it's 2/3 full. You already need a second freezer if you only have one. That goes for -80s as well as liquid nitrogens. The liquid nitrogens have been remarkably reliable, but it can go wrong. The alarm system can fail. The other thing I think is really important is the communication with the people who actually procure the samples. And it's very important to talk to the PIs, yes, of course. But I have found that I get a lot of mileage from talking to the people who are poking needles into the patients. The clinic staff, the nurses, the people who actually package the samples. You need to have a line to them, email, fax, preferably phone call, phone number, something to get in touch with those people and have them know that they can get in touch with you. Has saved a remarkable number of samples from being misrouted or mispackaged and just lost. And so I think those are the notes I wanted to make. There's a lot of things we learn along the way that we kind of learn the hard way. But again, Dr. Ramirez covered that really well. I really encourage you to take her notes very seriously. But anyway, again, I'll answer any questions.
Okay, well, I think I sort of began in my earlier presentation of sort of finished up with some of the things that we were asked to speak to of how we how we have used tissue banks in our own research, and I'll just elaborate a little bit on that and add a few comments like Dave did. So, as I said before, I'm a translational veterinary oncologist, I guess, for a lack of a better term. In fact, in our work, we work pretty closely with Dave here and Dr. Bird's group, using the dog as a translational model for hematologic malignancies. So sort of really wrapped up in that is questions where the biospecimen repository in particular has been helpful, is sort of addressing this question of just how good is the dog as a model for understanding human cancers and for developing new therapeutics. So we know that histologically, that if you take a look at a lymphoma biopsy under the microscope from a dog versus a human, say with diffuse large B-cell lymphoma, which is the most common B-cell lymphoma in both species, that they're indistinguishable. Likewise for osteosarcoma. So how far does that similarity go on a biologic and molecular level? So some of the first studies that we did with tissues from our tissue bank was Affymetrix gene expression profiling in dog lymphomas, and that we generated gene expression profiles of over 100 dog lymphomas from the bank, and basically interrogated those with just respect to understanding the dog disease in and itself, and then as well as from a comparative aspect with human gene expression profiles, and that was very informative in that we took away from that on a molecular basis that there are some striking similarities, but yes, there are also differences that it certainly would be a stretch to say that they're a copy of being exactly the same disease. For example, we find that in the so-called ABC or activated B cell type of lymphoma in people, that dog lymphoma probably resembles that the most, and that in both the human and the dog, that the NF-kappa B signaling appears to be very important in both, but we also are able to glean from looking at the expression in the dog that the dog probably gets there in a little bit different way than people. So, we have to keep that in the back of our mind when we're, developing targeted therapies, that the most relevant target along that pathway may be a little bit, may turn out to be different in the dog versus the person. That being said, it has also been pretty interesting to see with some of the therapies in some of the B-cell malignancies, as far as how predictive the dog model truly has been. More recently, when Dr. Croce joined OSU, we began a collaboration to do microRNA profiling of a number of different canine malignancies, including mast cell disease or mast cell tumors in dogs, where we identified a microRNA that appears to be pretty important in the invasive or metastatic phenotype. So those are just some of the ways in which we've used the veterinary school tumor bank. As far as if you're thinking about how that there may be a role for the dog model in particular in your research, I think really, like Dave was talking about, sort of putting some time and effort into the front end of things is always, I think, always very helpful when working with just about any sort of core facility here on campus, that it can save a lot of time and effort in the long run, particularly dealing with with dogs is something probably new to most of the audience. So a little bit of additional back and forth as far as understanding the model and as far as what can be brought to the table that you may not have thought about, or limitations. Like I said earlier, that for example, lymphoma is really common in the dog. So if you're doing a study where you would like to be looking at a couple dozen or 100 biologic samples from dogs with lymphoma, that can be accommodated, no problem. On the other hand, if your interests are in, colon tumors, then we might be able to give you a handful, but we're probably not going to be able to do a whole lot more than that. Dr. Jackson also asked a question about essentially prospective collections or other services that we might be able to help with. And I guess I would just say that if there's anything that you want to do with a client-owned animal/veterinary species, that you should reach out to us either at the Biospecimen Repository or the Clinical Trials Office. Both myself and Dr. London, Cheryl London, who heads up the Clinical Trials Office, we sort of wear both hats and work very much in both areas. That just because we aren't doing something or have a particular sample doesn't necessarily mean that it would not be possible to do. Just because certainly the scale of our operation is is a lot smaller than perhaps others. If there isn't, there hasn't been a request or a demand for it, we aren't necessarily, doing that right now, but it might be something that would be possible to do.
And the panel will be happy to answer questions from any of you all.
A more general question, but since you're both users and biobank repositories, I am thinking of starting a project looking at gene expression. So for me, RNA and the quality of that RNA is extremely important. And I was talking to a colleague who had engaged already into a similar project, and he was actually doing a lot of the analysis of the data. And he went back and looked at data that had been published and publicly available already studies and raw data from his field was in cancer. And looking through, he started to realize that there were biorepositories that had really good RNA and others that had really poor RNA. And in some of them, more than 80% of the samples that they provided had RNA that could not yield very good results for Affymetrix or gene expression analysis. So my question is this, going into this and knowing where you want to put your money for buying this and who you want to contact in terms of biorepositories, how do you get a feeling for where is the really good RNA, which one is really good? And for the biorepositories, is there maybe a national program or something the biorepositories themselves do to sort of figure out, get feedback about the quality of the RNA that they provide so that they can go back and look at their SOPs or look at where things might be happening along the process? Is there any quality checks that are done by biobanks that might be posted when you're looking for these things? Because I haven't really been able to find that kind of information.
I think that's a really good question, and that applies to any subsample processing that we would do, actually even the primary processing to look at viability post-thawing. And there are obviously the variables we all know about between, variables between samples, variables between cases, based on how the sample was procured or just the peculiarity of that sample. But more to your question is absolutely, I will say, please give feedback. If you obtain samples from the Leukemia Tissue Bank and they are poor in quality in any aspect, we would absolutely want to know that so that we can change something. We follow very, very well established protocols as far as cryopreservation. There's nothing unusual about that as far as, the usual freezing and the freezing media and the controlled rate and all that. Even though that's uniform, the samples don't necessarily come out with the same viability. We do test that. The RNA processing has been, according to commonly used techniques, but we are always interested in improvement. If what I would recommend is if you have a study in mind, we can either provide the frozen samples directly to you, or we would be very happy to do the processing there, but we would maybe do a subset, and you tell us if that meets your QC criteria. The way we do it, though, we have recently gotten a qubit fluorometer, which is very sensitive for quantity detection. It doesn't do much for quality and integrity, so we're teaming up with the Genomics Shared Resource to use. They have a bioanalyzer, adjuvant bioanalyzer, and a Perkin-Elmer instrument that will help to assess the actual RNA integrity. And so these things are, in the past, I would say, a year or two, becoming more in the forefront because of the RNA-seq methodologies that more and more people are using. We are checking these things more and more, and where we find deficiencies, we absolutely want to correct them, and we'll take any advice. But we have used several different commercially available kits Right now, we're using a Mirvana kit, I believe, is giving us the good consistency, good reliability results. But the main point is, yes, please provide feedback. We will tell you everything we can. We don't necessarily post the results because there may be, it's kind of a hard thing to sample for. We might do, 10 here and 100 over there, but just as often we're giving samples to, in the cryopreserved form to the lab because the people want to do it in their hands, which is fine too. And if they do something differently, we won't necessarily know what that is, if that makes sense. But yes, give feedback. If someone else wants to address that.
Irina Bohimsky, Nationwide Children's. Just to echo what Federica said, I think also what's important is for banks to have a quality control. So we have a lot of tissues, but actually we have a lot of tissues and we extract RNA. And it happened one time that one researcher that we shared tissues with wasn't extracting RNA and was blaming on the tissue bank, which we know that our RNA and our tissues are the highest quality. So we have a certain tissue that we keep and we give we have plenty of. And we give it to that person and say, try that, and if that extraction doesn't work, then we can blame back on the procedure. It actually, the columns for RNA extractions will not give you a good yield. So the best way is to really do it with a Trizolam, very old method, especially for RNA-seq. But a lot of the researchers, it's very easy to say the tissue bank is the problem, the storage is not proper, because sometimes it's inherent in the disease that they're studying. So we study the placenta. The placenta is full of RNases. So unless the collection doesn't occur in the delivery room, you're not going to get any good RNA that is stable enough. So the same with tumors. It's part of the disease process. So again, we found that having one or some tissues that we can actually ask researchers who are not satisfied with the quality of those specimens to check their procedure was very helpful.
Great idea.
And more general, as you develop a use or something that you want from a biobank, whichever it is, I think as you talk to the biobank and apply for the tissues, it's really important to outline in your application or in your discussion what you need and the quality that you need. And we didn't do RNA, but we had certain things that we wanted to do in terms of time points. And when we approached the PRR, they didn't have the time points we needed. So rather than get tissues that we wouldn't be able, or serum that we wouldn't be able to use, we just brought that up at the very beginning. So I think it's really important as a user to outline very carefully and to discuss with the particular biobank what you need and that sort of thing, because that way you'll avoid getting things that you don't want or tissues that aren't high quality for what you want to do. So I think it's really important as a user that you outline that in your request.
So I just want to add something that my colleague mentioned. So he did go back to the 2 tissue banks that have very poor results, and they looked back and they actually found that it was coming from the collection point. And they found that there were some surgeons that were collecting the tissues, they were not putting them immediately inside the preservation solution. And so they went back and they actually changed some of the procedures at collection. So that turned out to be important for the tissue bank to monitor back the collection procedures that were happening in the operating rooms. But it's something that just sort of depends on the end user giving feedback. And I'm wondering if there shouldn't be something, because these samples are so precious, to have 80% of samples in a tissue bank that are not usable, to be able to have a system in place to be able to check as a biobank the quality of what you are receiving and have checkbacks. I wonder if this is something that we should be moving into, or if there is some concern about that, even as patients are giving consent for these samples to be used for research?
We do check every sample on the way in. That is post processing, and we're, I think in a way the leukemia tissue bank has the advantage that our tumor cells can be obtained through a simple Ficol gradient centrifugation. Every sample, as it goes into the allocation for whatever is specified, usually it's cryopreservation, the viability is checked at that point, and it's recorded, and we maintain all those data. And if we are freezing of a sample that has a very poor viability, we will absolutely note it, and depending on where that is, we may make the decision not to bother with it because the viability is too poor. But all of that is tested as the sample comes in. But it's very important. You're right.
Yes, I can address a little bit with respect to both our bank here as well as the greater CCOGC effort. So, it's sort of that whole question of QA/QC, both going in and going out, and where do you put resources, particularly in a smaller operation such as ours, or even CCOGC, which is centralizing collections from 6 other sites, is still relatively small, and at least for CCOGC, with the original thought was that there would be a fairly extensive assessment of every sample that went into the bank, with the funding support that effort had, that really was determined eventually not to be feasible. So what can we do to assure excuse me, to assure quality the best we can, and what was really decided upon there was a subsampling of about 10% or so of the samples in the bank, covering all the different histologies, and at least to be able to say something about the overall quality of the samples in the bank, and then ultimately leaving it to the end user as far as if they want additional assessments done either directly through CCOGC with partners, or that they do it themselves, that it was sort of on them as far as the assessing the quality of the final specimens, and that exercise turned out to be very useful. That we were able to come away with realizing that the samples in that bank overall had very high quality, but also in the course of that identified two problem areas. One, that exercise told us that one particular histology was very highly prone to collections of tissue that were not going to be of sufficient quality to for most applications downstream. That tumor type being hemangiosarcoma, which in the dog often occurs in the spleen, and it's very difficult for the person making the tissue collection to necessarily be able to identify tumor tissue from hematoma and other tissues there. So path review is especially important in that particular histology. It also identified very early in the collections of one of the 6 sites that just was sort of mentioned in another context there, that there was poor RNA quality from a particular site in a particular time period, which was eventually, again, tracked down to the early SOPs, and that, the time from when tissue was leaving the patient and getting to where it needed to be processing, that there was a problem there that was later rectified. We know within I've used this, the bank myself quite a bit, and so we have quite a bit of knowledge as far as that. In general, our samples seem to have, collections seem to have performed very well in various downstream applications. But we're in the process of doing sort of, although we parallel collect for CCOGC, we're doing, having an effort for sort of a similar sort of sub-sampling for our particular tissue bank overall as well.
Do you have any experiences with the IRB approval process that I want to share, or hints for success or anything like that?
We have actually a lot, and it's been, I think they've been really helpful. There's a fairly complicated, I shouldn't say complicated, it's what we're trying to do with the Leukemia Tissue Bank is anticipate what investigators may want to do. So it is broad, and that I think understandably lends itself to questions about, where are the limitations, et cetera. But we don't give out identified samples. They are, in this case, coded, but the investigators who get samples from us never get the code. So for their purposes, they would be de-identified, the investigators who receive the samples also have IRB approval to work with de-identified samples. And I think we've worked together really well. And I haven't been on the in the directorship that long, but the last time it went through, I think it went through very well. I don't know of any particular issues. I think there is a general carefulness. I mean, we have all these policies in place to make sure that the rules are followed and all that, and I think that served us pretty well.
For the perinatal research repository, we had a little bit different issue because we had collect samples both at Children's and at Ohio State. And one of the really nice things that's happened over the last several years is that Ohio State's IRB and Nationwide Children's IRB work together. And so you really only have to get approval from one of the IRBs, and you submit to each IRB and they decide who should be the controlling IRB. And that's really helped us a lot. So we actually collect samples from women at OSU in childbearing age. They're not children, some of them anyway. And we do it based on a Nationwide Children's IRB approval with OSU consent or OSU assent or OSU IRB's assent. So that's really nice that they work together because that helps us to do things that much quicker and that much easier. And so that's a really nice thing that's happened, I think, in the relationship between Children's and OSU.
Just from sitting on the NIH, it actually helps a lot having a boilerplate write-out for resource sections. At NCH, we have one for the Perinatal Research Repository, but just to emphasize that our institution has biorepository expertise, even if it is not necessarily for the really small niche, and we still have human subjects. Just the fact that there's something to grow upon and a culture, it helps a lot. Mm-hmm. So I think it would be useful at the end of this workshop even to come up with some shared knowledge about biorepositories at Ohio State NCH that are available and are accessed. For example, I'm interested in positive control tissues from multiple myeloma. So, instead of really buying them, I think it would be much easier to access one of the banks.
Absolutely, and we would love to help. I think the earlier comment is that we don't matter or exist if we don't have users. So we want people to use these tissues. One of the things I would like to take on is making the initial contact easier and more streamlined so that people can learn more immediately instead of trying to track down the right route to learn these things. And yeah, I think your point is very well taken.
Yeah, I think that my experience with investigators, especially with the cancer cooperative groups, have been that people are, investigators, especially young investigators, they have in mind what they want, but they don't know where they have to go to get the tissues. And I feel really sad for those that waste their time, sometimes going after specimens with clinical annotation when they really don't need that for their study. And I spend more time on the phone telling people, this is not the right bank to ask for tissue. You should go and get the CHTN or get something else. So considering the number of resources that both institutions have, I think it will be a good idea to have some sort of resource in which people can actually go and not just says, for example, OPRN is nationwide. These are the types of tissues. It doesn't have to have, I don't expect anyone to put down their inventory, but at least say, these are the types of specimens that are available. You have, blood, plasma, soft tissue, and then it comes with this or that clinical information. And then people can at least have an idea as to what is available out there in a resource. It could be something online, which people can go and have a little summary of what is it that each biorepository does, or what is the CHTN, what the pediatric CHTN does, because I think that will be very helpful. I really think that investigators waste a lot of time trying to find the right resource unless they have a mentor or somebody that can tell them just go, or, I hear a lot of call Nilsa, she'll help you. But, at least have somebody or some way in which we can go somewhere and find this. I think it's very important to put those resources out there so our investigators know what they are.
Yeah, I agree with you completely, Neil. So I think the biggest barrier that I see for junior faculty is figuring out who to call, who to contact. And so having a central repository or a central place where they can get data on what's available, I think would be perfect.
So Rutain from Nationwide. So when you have a central repository and an investigator from one of the centers that has contributed towards that repository wants to study, wants specimens, what are the processes in place to decide if their science is robust? Do you have a scientific review board? How does that work out?
For the perinatal research repository, there are 5 people, a couple of OBs, one neonatologist, and a couple of PhDs who look at. There's a 4-page application or 3 or 4-page application, and they look at that to see if it's if it is okay, and if not, they suggest revisions and questions and that sort of things. And the questions they ask are pretty general. Why do you want them? What are you going to do with them? How does it fit with what the OPRN is doing, and that sort of thing. So, and I think most tissue banks have some sort of thing like that. But there's a group of people, almost like a review board, that decides, and they work pretty quickly again, because it's, at least with the PRR, it's a fairly short form, and so they can do it pretty quickly and give you feedback. And they again, biorepositories exist so people will use them, and so we want people to use them. So we actually try to, if a certain proposal may not be, I think you used the word robust enough, we'll try to help the investigator make it robust enough so that we'll feel comfortable using the tissue, we'll feel comfortable giving them the tissues to use, because we really want the repository to be used.
The LTB has the same exact thing. We have a steering committee that goes through the proposals. They're fairly short, it's fairly informal, and we'll provide feedback if it does not meet criteria, how we can make it meet criteria, exactly what you said.
I just have a quick question about information management systems that you use in your labs or in your biorepositories. If you could wish for something and make it happen tomorrow, what would that be?
That's a very good question, and I think that we have a bit of a line that we straddle as far as being very, very careful with patient information, PHI, confidentiality, and what was talked about earlier, with getting the investigators what they want. And investigators, I'm one, we want everything. I mean, no, we don't need to know the person's name, but if you, everything that the clinic knows about this person could potentially be interesting. The policies that we have are stipulated by the IRB and the NIH rules about exactly what constitutes what kind of information. We record the basics, that is, cell number, viability, and all, just in the coded identifier. All that is easily available. We just have it in a database called, you might have heard, a database called TransChart. We have the ability then to link certain people within who have, passed their city training. Actually, all of our staff has city training and all that. But the certain subgroup of our staff can access clinical information on the honest broker policy. That is not simple. It's not streamlined. I myself actually don't do that. I'm technically allowed to by the IRB, but I don't do that. So I'm not familiar with all that it takes, but it would be really nice if there was a way to broadly search everything for everything at once, rather than searching for what samples and then taking that information and going over to the clinical side and getting that. But I don't know that that's possible without compromising, I think, the very, very legitimate concerns of making sure that the protected information stays protected.
What about on the back side? Like, you send their samples out, they go to wherever, the researcher does their research, and then do the results of that research or those analyses, do they ever come back to inform the sample, I guess? Is that,
They can. We have instances where people are doing maybe a mutational analysis and they will share the results, but that's not part of our requirement for supplying samples. So it's more of that would be a collaboration that we would be very happy to enter into, but we don't require it.
would that metadata be ever helpful for the next researcher?
It could, and we could introduce the possibility of investigators sharing what they find for other people to access. They may or may not be, reluctant to do that. I don't know how that would. I haven't tried that. I think that's an interesting idea as far as how it would go over with the investigators doing the work. But I look at it as they receive the samples under the agreement and the IRB specifications and all that. They're not compelled to return their information, but I think if they did, yeah, we could keep that and potentially make that sample more useful down the road. I think that's a great idea. If you have comments about that.
Other questions or comments? Now's your chance.
I think we just to finish up, if anybody has any other questions, I think all three of us would be very happy to talk either after this or by email or phone or whatever. Like this is actually kind of fun, but please don't let the time here be an obstacle to contacting us and getting more information, because we do want to expand our user base, absolutely.
Well, thank you very much, panel.